Sequencing is not about DNA. Just like the PC
Right.
was not about one use case,
Right.
right? Many different use cases mapped onto the PC. In the same way, all kinds of molecular testing are going to map to sequencing. So DNA, RNA, proteins,
Protein.
lipids, metabolites, structural information, spatial, all kinds of things that you don't even think have anything to do with sequencing today are going to get mapped to sequencing.
Okay, so that just makes the beginning of a new episode. Thank you so much for coming. This is actually a series of episodes we are planning to speak to industry professionals from our diagnostics and biotech field. And the idea is to have a free flowing conversation that people from, again, our industry would benefit out of. Call it tech, call it the depth of tech that's coming out in the future. What kind of waves are we sort of riding today as a whole, as a country, as a world in the biotech sphere? And also a little bit about what's happening with your company, and what are the exciting things that are going to come out this year and further. So the conversation will be super free flowing, and we'll take it as it goes and try to have fun with it.
Sure. Thanks,
Okay,
Rohit. Yeah, very happy to be here.
thank you. So before we jump into the mainstream things, I'm sure you had a very eventful year so far. How has this week been?
Yeah, so it's been a very exciting week for us because we have sort of done a preview of our second product to a few select customers or potential partners, and gotten into those kind of partnership conversations. And some of those conversations have really, come to a very exciting stage right now where people have expressed interest, things going ahead. So yeah, so this week has been, uh, yeah, I would say in some sense, internally some kind of validation for our vision, some kind of validation for the work that we have put in over the last 3 years. So
Right.
in some sense, it's a good time for me to be talking with you right now. Yeah.
Oh, great. Awesome. So now that we are in it, tell us a little bit about AlgoBio. Where do you come from? And what has been up so far?
Sure.
And we'll go into the details.
Sure. So actually, even before AlgoBio, it may help if I give you a bit more of a backstory.
Right.
Yeah. So I mean, growing up, I've always been fascinated by science fiction, Isaac Asimov and robots. And I guess Asimov mainly, and then robots and Foundation series, both of them, right?
Right.
And somehow there were two themes that stood out for me. One was computing and what happens when computing leads to AI and intelligence. And the other was the Foundation series of something called psychohistory, by which
Okay.
you can write down a bunch of equations and predict the future of humanity itself. Asimov was a chemist, by the way. So
Okay.
according to Asimov, it was like statistical mechanics applied at
Okay.
human psychology level, right? So that's why he called it psychostatistics. And so I was fascinated by these ideas. I was going to start graduate school. When I was going to start my PhD, I decided to work in this field of research called molecular computing.
Right. And this was
And
in India.
this was
No.
in Los Angeles,
Los
University of
Angeles.
Southern
Right.
California. My reason for choosing this area at that time was I was very excited about AI, but I wrongly, in hindsight, felt that the statistical approach to AI was not going anywhere and it needed a more fundamental rethink of what is computing.
And
Yeah.
this was roughly the year
This was around 2003.
2003. Right.
Yeah. So anyway, I went into this field of molecular computing, and I it definitely did play into my interest in both computer science, computer complexity theory, and computer science theory broadly, and these ideas of emergence and non-equilibrium statistical mechanics and so on. So I got to explore both these directions. I got to work in a lab, do experiments myself, and so I think it connected with some of my uh, like my need to understand the world at that level in a very nice way. So it was quite fulfilling in its own way. And so I continued, some of the problems I started during my PhD. Uh, in some faculty positions later on at Duke University, TIFR, IIT
Right.
Bombay. So yeah, before I knew it, it was like I had been about 20 years or so in doing research on this
Right.
kind of. So this field of molecular computing, the key idea there was, can we build a computer that dissolves in water, right? A computer in a test tube, and
Computer in a test tube.
a computer
Right.
in a test tube,
Right.
right? That it dissolves in water, and when dissolved in water, it is able to perform computations.
Right.
Yeah, and so of course, so one question is, why would you want to do such a thing? And the kind of fundamental science answer to it is that it is a model for what a cell could be doing, and if you
Correct.
could build interesting computation within a test tube, then perhaps the kind of mechanisms you reveal that are effective could lend some insight into how cells might be working.
Right.
Yeah, but that's a scientific answer. One would also like a technological answer. What can you do with this, right? And so I would say that there hasn't been a good technological answer for a very long
Right.
time, right? What do you do with it? Nobody really had a killer app
Right.
with molecular computing. Yeah. So for AlgoBio, the birth of AlgoBio sort of happened during the COVID pandemic, and partly it was a realization that these kind of ideas, the molecular computing community and the way of thinking about computing in a test tube, um, really has a very big role to play in the extraction of information
Right.
from the molecular scale to the scale of you and me, or the scale of the
Right.
computer, right? And so that insight really was the birth of AlgoBio, and the name itself, right? So it was chosen to reflect algorithmic biologics, right? So
Right.
biologics, of course, are the molecules that are commonly present inside cells,
Right.
and we wanted to indicate that somehow thinking about the algorithms that can be performed with these molecules uh, is going to lead to a whole new way of looking at many
Right.
existing technologies
It's
and so on.
actually one of the very thought through names that I've come across. Because like I've been in the space in the startup game for a while. Sometimes people sort of say that name is what you make of it and then move with it, right?
Mm,
But in your case, when the first time we met and you explained the whole logic,
mm,
it was super interesting. Like you can pretty much directly sort of understand what you're up to with just that name.
mm.
So that was like
Yeah.
very interesting
So,
thing
so
that I
I mean, to
noted.
return the compliment, I
Right.
love the name Cambrian
Right.
Bioworks as well. I think it's a very sticky name. I think you don't
Right.
forget
Did
it.
I did
Yeah.
I tell you like how that came about?
No, no, I
So this
haven't
is
heard it.
actually Vaibhav, my co-founder.
Uh-huh.
There is this era called Cambrian explosion,
Right.
prehistoric era,
Mm,
where sort of biology evolved into
mm,
multicellular organism and dinosaurs came, everything else happened.
mm,
So that era was like a steep growth curve.
mm,
And our idea was okay, we are in this space, COVID happened,
mm,
we built the company.
mm.
The way we were moving at the rate at which we were moving at that point felt almost crazy
That's
in every level.
fascinating. Yeah.
So that's the idea. So
Yeah, but
Cambrian
not just that, right?
explosion,
Yeah, I do think that there is a Cambrian era coming from bioengineering as a
right,
whole,
right,
that we will see kind of
right,
an explosion of some kind for the number of use cases and so
right.
on. Yeah.
So coming back to your original insight around building AlgoBio, right? Tapestry happened during COVID,
Yeah,
or
it
just
happened.
about that time, right after
Correct.
that.
Correct.
And did you start the company after that or
So,
before Tapestry
yeah,
happened?
so in 2020, when we first started hearing about COVID, uh, so I realized that I had something to contribute. Uh, I was fortunate enough to find some good partners on the biology side who could help me, with getting the experiments off the ground and so on,
Right,
actually at NCBS Bangalore.
right.
And so with that, I think we very quickly got a paper out where we had done the experiments, things had worked beautifully. And
And this was still before registering a company.
this was all, yeah, I was in academia, I
You're
was a
still a
professor at
professor
IIT
then,
Bombay.
right?
Yeah. So when this paper came out, it had a, like, till that point, I was trying to solve a problem for me. After the paper came out, we got a huge amount of press, lot of attention. But then actually getting change done in the world there were so many hoops, right? Getting the kind of even though everything was accelerated in COVID, because I was so new to this game, I did not know that I didn't even know that there existed something like, drugs comptroller,
Right.
yeah, DGC, yeah,
GCI,
General
yeah.
of India
Yeah.
or CDSCO. I didn't even know they existed. So I was in that kind of a world, right? And so when I realized the challenges that were involved, and then it really hit home to me that, hey, I'm building technology, but sitting in academia just building that technology, I'm not going to change the world within the kind of time span that I want, and I'm also not going to get the resources that I need to make this a real solution.
Right.
And there is a gap between what I'm going to build and what is required to play this regulatory game and then
And then the
actually get to
commercial
people.
game eventually.
Yeah. So then I started, understanding that. At the same time, I also realized that the technology that we had deployed, that we had sort of proved here, was not a single case. It was
Correct.
not about COVID. It was more fundamental than that. It was actually a completely new way of thinking about pulling out molecular
Right.
information. And not only that, it tied very well with my background in molecular computing,
Right.
because molecular computing had a unique role to play in the way that we were envisioning this field. So, and then I realized there were a couple of other problems which were commercially very attractive, where this kind of an approach could be very effective.
And this realization was more towards the second half of 2020 in terms of timeline,
Yeah, second half of
towards the end
2020,
almost.
right? So then in my mind I was looking for these opportunities. At the at around that time, one of my students from IIT Bombay, who had been working on this project with us, she graduated, she came to Bangalore. She met Nidhi from Axilor,
Right.
and so she was actually part of this entrepreneurs first, or one of those entrepreneurship programs. So Nidhi was a mentor at that program, so she was telling Nidhi about her startup idea, but also ended up telling Nidhi about what she was doing before. So we came into the story that way, and Nidhi got intrigued with what we were doing, and so she ended up making an introduction. We had a phone call and hit it off, because Nidhi had sort of, for me, the missing side of the puzzle, right? So I had the tech, I had the vision there. Nidhi knew what it took to build some kind of a startup
Zero
in
to
this
one
space.
story,
Yeah,
essentially.
and everything that came in there, right? So um, then, when I realized that there was genuine interest from investors, actually there was one more investor who got interested. We got two term sheets, ended up going with Axilor, ended up going with Nidhi, and then moved forward from there.
So
Yeah,
that's the beginning of your
yeah. So that's how the
enterprise,
company
essentially,
started.
the startup and
Correct.
first small team. 2020, it's what, 4 years now? 2020, 3.5 for you since
Yeah,
you've begun.
yeah,
And
yeah,
how has it been? Like,
yeah. So it's very different. So I would say that in academia, in research, you have to work very intensely, and at least that was my way of, I'm not saying everybody works that way, but for me, I would work very intensely, uh, not like 12-hour days, but the amount of concentration I needed and the kind of state I needed to get into before I sat down to do some work
right,
it was quite challenging. Yeah. uh, in the entrepreneurship world, I think it's it brings, there are a lot more meetings, a lot more sort of uh, discussions, and the challenges are somewhat more subtle in some sense, that in academia the problem is in front of you and you have to go and attack it, right? So here, you have to realize in the first place that there are problems, and you have to, sort of uh, so of course learning has been universal, I would say, right? So there has been learning.
of
When
course.
you're an academic, you have to keep
Right.
learning. Here you have to keep learning, so that investment is the same, but lot more uh, people work. And what I found, what I discovered was that I enjoyed that a lot. That
You mean
I'm
the entrepreneurship game or the focus
people
in
work.
people
so
work?
so
Right.
I think academia for me, right, it depends on personalities, right?
Right.
For me, academia got very lonely at times.
Mm.
uh, but yeah, I have found that having people around and having the opportunity to bounce
Being
ideas
being part
off.
of a team and building a
Correct.
team and having
Correct.
that team
Yeah,
around you.
that
Right.
works very well for me. So
This is an
that has
interesting
been,
question. A lot of people ask each other, or ask during when they sort of building stuff.
yeah. Hmm.
Are you a team player or are you an individual player? And not everyone seems to know what they're
Yeah.
better off at.
Yeah. Yeah. I
I think
don't
it takes a
think
lot
I
of
even
time.
thought in those terms
Right.
while I was in academia.
That's an interesting thing because I kind of realized something similar after whatever going through building companies
Mm.
that for the longest time, although there was a larger team around, as a startup, as a smaller startup, as a team with say 5 people in a startup, everyone does everything. And within their context, they're pretty much lonely because when they're tackling a challenge, they're probably attacking end to end of that challenge. There is no junior or a team, or there is no management as a system. There's no hierarchy.
Mm.
But later on, as I evolved and we sort of grew as a company, team wise, number of problems we have to attack wise, once I realized that you have a team that is dedicated to say a department or a broad theme of issues, and then there are 2 3 people trying to solve that broader theme of issues, I think life just becomes a lot more fun
Mm.
by default.
Mm.
Like instead of you solving that one problem or one large thing, 3 people are sort of working on it together and you evolve, creative stuff happens.
But I also
So
know people who would rather be left alone to work on it.
which
So it
is true,
depends on people.
which is
Yeah.
true. So I realized as well, like you said,
Mm.
being part of a team or a teamwork scenario is a lot more fun for me personally,
Mm.
and only learned it way later
Mm.
than people should know, I guess.
Mm.
So that's awesome. So coming back again to your tapestry for a bit before we jump into the future.
Mm.
So you've already built a COVID test uh, where you could pool multiple samples and give results independently, including the quantitative metrics of each one of those patients, right? So can you tell us again, like I said, molecular biologists are watching, entrepreneurs are watching, primarily players in our clinical diagnostics world are watching,
Mm.
sequencing genetics world are watching. So maybe tell them a little bit about how the system works.
How Tapestry works. Sure. Uh, yeah. So um, the kind of big idea here was that how do you, like how much information do you need to get out if you are going to test a large number of samples? Say you're going to test 1,000 samples. Okay. Um and say I am doing the testing, I have done the testing. Okay, so I have all the 1,000 results. Now I have to send you a message. Okay, I have to tell you, let us say forget CT value for now. Okay,
Right,
I have to tell you which samples are positive.
right.
Okay, and imagine that we know beforehand that no more than 10 samples are going to be positive.
Because of
Okay,
the general prevalence rate and whatnot.
yeah, so grant me this. Okay,
Right.
then how should I send you a message so that I have to send you, so that I can send you the shortest possible message? Okay,
Okay.
so this is the kind of question that is studied in coding theory or
Right.
compression theory, right? And the answer turns out to be that you don't need to send a very large number of messages. So to give you an extreme example, if there is only one person who is positive and we know beforehand there is only one person, I only need to, answer questions about whether the person is less than 500 or greater than 500, and you know in the in which quarter, in
Right.
which eighth, in which 16th, and so on, right? And by answering some of those questions, right, and then finally order even like 10 questions, right, 10 yes no questions, I will have told you exactly which person was positive,
Pointed out the
right?
person. Right.
So uh, what if the test that we perform, instead of telling us whether one sample was positive, could give us the answers to these kind of questions?
Right.
Okay, so if we could arrange the test itself to tell us, the one bit that the test is telling is not whether sample 3 is positive or negative, or sample 7 is positive or negative, but whether an odd or even sample is positive, or whether the sample that is positive is in the first 500 or the last 500, or in the first and third quarters or second and fourth quarters, right? So what if that could be done in the test tube, right? What if this kind of compression that I'm doing when sending you the message could be done in the test tube? Okay, then the number of tests you need to do becomes much less. It's
Based on
the
number
size
of,
of the compressed
right,
message, right?
right,
So you don't need 1,000 tests, and you could imagine that you could do it for much less. And in fact,
right.
if you have like a 10 out of 1,000 positive, any 10, we don't know beforehand, this reduces to a message of size some 100. Okay, and so in 100 tests you should be able to give 1,000 results.
Within 100 tests, you pretty much
You should be able to give
tell
1,000 results,
all
right?
the 1,000
Yeah,
yes or
so this
no's.
is a purely non-constructive uh, thought experiment.
Right,
Okay, that it is possible because of what we know from information theory,
right,
from a basic counting argument, right?
right,
And now to make this practical, you have to decide what are the questions you are going to ask beforehand. You have to decide without knowing who is positive, right?
right,
And so the questions that we are asking are the different uh, pools that we combine these samples into. So
right,
each, right, if I could make a pool out of all the even samples, pool out of all the odd samples,
right.
if
Fair.
the pool out of even samples is negative, then no even sample is positive.
Correct.
If the pool out of odd samples is positive, then at least one odd sample is
Right,
positive, right? So of course there are other limitations coming from the reality of the lab and molecular biology and so on. You don't want your pools to be too large,
correct.
right?
You
You lose
can't
on
fill in
sensitivity.
all the
And
samples into a
yeah, and you
larger
also don't
tube.
want your pooling scheme to be too baroque. You don't want
Hmm.
like samples going all over the place in some really complex patterns. And you also want a practical decoding scheme. Once these pools are tested, once you get the results, you want a way to go from the pool results to the individual results. So
Right,
you need an algorithm, and for that algorithm to have good performance, you want your pooling to have good performance.
right,
So these considerations led us to a pooling scheme. And that pooling scheme we worked on very hard to make it very easy to implement in the lab. And this was also, a learning over the course of time. The very first pooling scheme we did was mathematically perfect
right,
and completely
right,
useless in the lab.
right.
So we later went on to pooling schemes that kept the goodness of the
Realities,
mathematics,
right.
but at the same time much easier to perform in the lab.
What about the sample input volumes, right? You only have a limited amount of sample volume per patient, and you can't be splitting them into more than X number of realistic pools. So keeping that in mind, let's say for a 200 microliter or a or even a full sample, which is a 5 ml tube, EDTA tube, there must be a theoretical limitation, right, to how much you can pull. So do you already have that answer?
So in the schemes that we have been commercially kind of deploying and so on, one sample goes to three pools.
That's good enough to ensure everything is okay.
So one sample goes to three pools, you have enough
It's
samples,
like
right?
one third,
Three, yeah,
right?
three ways is usually okay, right? And you of course know this better than me, but at least during COVID times, we were, you know, we were collect the compared to the amount of sample that we were collecting, the amount we were using for testing was like a thimble from a bucket.
Right, of
It
course.
was a very
In
small
in
fraction.
all
So
pretty
so
much most cases also.
yeah, so then we do,
Unless they're doing
yeah,
multiple tests with
yeah.
it, fair. Then
Yeah,
it's a
so
different
so
story.
it didn't really come up for us from that direction that one sample cannot go to more. It
Right.
came up to us from the more practical direction of completing the pooling and making sure that one pool doesn't contain too many samples. Because if
Correct.
one pool contains too many samples, then you have to worry about, do you have enough of each sample in here? Do you have the sample volumes? Will it work on the existing kind of qPCR setup and those
Right.
kind of
So
things? Yeah.
essentially now it's come to this one in three into one.
So
Three different
one
samples
sample
into
goes to
one.
three pools.
One sample goes to three pools, which is as good as saying one third is the maximum you'll split a single incoming sample.
Correct.
Great. And then you do the extraction and
Mm,
then you do the whatever, either
correct.
PCR or NGS, and
Correct.
essentially
Correct.
awesome. And this was already achieved in 2020 and you already had data.
Yeah. So this was achieved in 2020. We had data. The initial schemes, as I said, right, used some fairly baroque pooling schemes, which were a pain to carry out in the
Right,
lab.
right,
And I had some, I had the good fortune to work with some really good graduate students who, managed to pull this off. But then when we started going to real labs, it became very challenging in a commercial
Right.
setting, right? Day in and day out to do this kind of pooling within a short period of time was too challenging. So then also we saw some experiments where there were mistakes that could only be explained if there were some manual mistakes, right? Manual
Typeting
errors.
errors.
And it was of course too challenging to do this, right? So then we started thinking about this, really simplified that, came up with a pooling scheme that could be carried out with minimum number of pipettings and without requiring an intense concentration from the experimenter. Once we got that in place, then people actually started using it, and yeah.
Right. And this technically could have worked with any extraction and PCR kit as long as your algorithm is used
Correct.
to compress and
Correct.
decompress.
Correct.
Right. This is something I very well remember from
Mm,
our conversation at that point.
mm,
So where are we today? Like started there,
mm.
and what are we seeing?
Yeah. So there were a couple of realizations, right? A sort of big picture real. So one was that this is not about COVID, as you said, right? It could be
Right.
about any other qPCR kit. It's not even about qPCR. This could be about mass spectrometry, next generation
Right.
sequencing, and in fact we have extended to various platforms
Right.
including mass spectrometry, next
It can
generation
be even
sequencing.
ELISAs or CLIAs.
Correct.
Why not?
Correct.
Right.
So it can be various kinds of tests, right? And the third thing is that this is not even about pooling. Okay, this is not about many samples pooled together, right? The way that we are using pooling here is essentially to compute answers to certain questions, which are more kind of global questions rather than
Right.
local questions.
Right.
And that requires some computation, right? And that computation we are achieving by doing pooling. Okay. But in principle, this can work with a single sample for many target detection. And again, to detect many targets, we can do some kind of an assay which will perform this computation for us, and we have built this in the lab today, right? So
Right.
we now have assays that are able to take a single sample and test it for a very large number of targets in a very small number of reactions.
And that's the exciting new part,
That's
right?
the second product
There is
that,
another
yeah.
industry heavily going upwards in terms of how many assays they can multiplex within existing systems. Again, your typical PCR point of care devices, right? There's a big world which is showing promise on more colors, more packing density, more targets within single tubes. You're talking about ddPCR now going
Yeah.
multicolor. So the excitement is towards being able to get more info with as minimum or limited number of tests that can be performed in as quick setting as performed as required. So keeping that world, we all know it's moving, it's changing. So how are you planning to sort of integrate? Are you also building hardware? What's the plan? How are you going to go attack that side of the industry?
Yeah. So this is a go to market kind of a question, right?
Right.
See, what we have is an assay platform.
Right.
Okay, we are able to build many different assays. So if we choose one area, say we say I want to build for AMR sepsis, right?
Right.
Then we will spend our like lot of time, money, and effort on that. Yeah. Um, we have an expertise which is in the molecular science, and which is in this kind of algorithm space,
Right.
right? And so we want to stay with our expertise and get into the right kind of partnerships which will allow us to get these kind of products to market faster, and with partners who know the business, right? Who already
Right.
know what to build, and how to distribute, right? So they know
Exactly.
who the customer, and they know that what the customer needs, right?
Right.
So this kind of deep market knowledge will take us years to gain, right? Talking to clinicians and then understanding the clinical situation and then understanding the existing products. So this is not, and then we'll have to just focus on that one area and do all of this. So it's a full partnership model that we are doing,
Right.
where we are telling we are going to multiple OEMs, multiple assay manufacturers, and we are telling them, hey, we have this cool technology, and what can we build for you, right? And how can we take this to market together? So that has been the kind of.
So then we can probably talk about an announcement that has already been made, which is again congratulations on that. Your
Thank you.
first larger fundraise. Uh, this, would you call it pre-series or
Yeah, this would be a
essentially a
pre-series. Yeah,
pre-series?
yeah.
So maybe tell us about that. How was the journey? How hard was it?
Sure, sure. So I think so we were of course hearing that the market was very bad and that was what was coming out.
Yeah,
So we were fortunate to end up with three term sheets,
right.
and I think something went in our favor. Possibly, so as a founder, I am not so yeah, I have not been very long in the business world. This is my first startup, right? So on the tech side, what we had, I think, was a fairly unique kind of idea, fairly unique kind of concept, and we had some early traction. We had very strong relationships, and so I think we were able to create that buzz. We were able to create that excitement in investors, and also when they went for the second level of vetting, when they were talking to our lab partners or with other potential customers and so on, they got back very strong feedback about the core technology, about the core product. So I think that worked big in our
Right.
favor. Yeah.
And this is something to note, right? In India, at least in the kind of situation we are in, which is starkly different from what happens in a San Diego scenario or California. When generally West life science startups, again, very different because most founders, with no offense to anyone here, have tended to be relatively more non-tech in India, at least in our space,
Hmm.
or distribution turned into product, or originally commercial players who sort of tried to get into product. That's what the system has been so far.
Hmm.
Only now, I think, in the last half a decade, is when we are seeing deep tech startups come out of academia, where people who've been in the field coming out and sort of starting companies is happening. I think your case is one of those very few where you've spent a lot of time in research firsthand. You taught, you're a professor, you are a deep tech person as a professional, right? And then you sort of became a founder. And this, I don't see a lot happening in India. Why do you think so? Like, why is that different for you? And
I wonder, I don't know actually. Um, I think that possibly it is, some kind of a pipeline or some kind of a, lane that needs to be created, right? So people need to see one or two people doing this and it will happen, right? Uh, there have been, if you look at Shanti Swarup Bhatnagar or many of these famous scientists, right? Even Homi Bhabha or, lot of these people, if you look at their careers and look at what they have built, right? They were very entrepreneurial, right?
right,
Uh, and somehow exactly now that the meaning of entrepreneurship has changed, the
right,
lane of, right? So now there are venture capitalists, and now entrepreneurship has, the game is a little different than what it was in their time, right? But that embrace has not happened, I would say, right? So out of there was a time during the startup boom when India was talking about like 100 unicorns or something like that,
right,
right? And that list of 100 had very few that had come out of academia,
right. Pretty much,
and
yeah.
yeah, or
Almost
deep tech academia.
very,
So
very
so
small
yeah,
number.
so I think um, somehow there has been a gap in terms of academic research coming to market. I don't know what to make of it. I don't know
Mm,
why. uh, when I started off, I tried to invite a few of my colleagues who had worked
right,
with me on this Tapestry project to also come and take it up, uh, like be co-founders and take this forward. um, it was, I think it also has to align, right? In some sense, for me the vision was a good fit. It was a mission for me, right? That this is what I've been doing all my life,
right,
and by finding a killer app for molecular computing, I can bring this at scale, I can make a real impact on the world, I can get sort of the full impact of the research I have been doing, right? Not just writing papers and getting few citations. So it made a lot of sense for me. I saw it as a continuation of my journey. Maybe
right,
for the others this was not that natural continuation, it was an opportunity in a time of crisis to make a contribution. uh, and yeah, but I do hope that many such startups will
Yeah.
come out of it.
Before we started this, right? You said something about the marketing stuff will begin next year, and this year is all about putting your head down and building the product. So, what are you planning? Like, are there any exciting things that you're looking forward to for marketing? Or this is again, think of this as advice for future founders like you who are turning research into tech, tech into product, product into a commercial venture, and then eventually going into distribution at some point.
Mhm.
Are there any things that you would sort of advise them and also tell yourself in the process?
Well, I mean, so one thing is that I think especially for tech tech founders, right? People coming from a technology background, I think there is a blind spot for how much learning you have to do. Uh, because I think there is a tendency to think that we have engaged with very complex things, and so the world of business cannot be that complex, and it is
Right.
something that we will anyway figure out.
Can be
Okay,
can be figured out, right?
it can be figured out. That's not false,
Right.
but just realizing how much learning there is to do before you even start to figure it out, right? That is, I think, something that tech founders especially tend to underestimate, and having somebody from the business side, at least in your early days while you are still learning the ropes, is very very useful, saves you a lot of time and kind of puts you in a place where you can you avoid some of those big mistakes you would otherwise have made.
Right. And then there are other broad issues as startups, right? One of the most hardest, I wouldn't say it's a problem, but it's a skill that we should all sort of build, is hiring,
Mm.
right? And in our space, in our industry, molecular biology as a whole, I'm talking about the biology side of things, right? Not the algo side of things. How has it been for you? Has it been easy, hard? What was your winning method
Yeah,
when it comes
yeah.
to
So
hiring?
I think I would put it this way, right? So hiring is one bucket. I would say that running a business, there are some at least 20 things or maybe 100 things depending on how op savvy your business is, where you have to be very rigorous, right?
In
And
every
rigor
aspect.
rigor looks very different, so, and I have a clear definition for rigor, right? This is something that I have kind of realized, that if you are doing the job, any job, right, say hiring, the way somebody who has just passed 12 standard would do it, then you are not being rigorous, right? So
Yeah,
what are you adding to it? And a fairly smart person who's passed 12 standard, right, what are you adding to it to make it rigorous, right? What processes are you bringing in? How are you making sure that you are delivering quality there, and so on? So I think that extra layer is very important to develop. It's a muscle to develop.
right.
And so I think hiring definitely has been a muscle that we have developed over a period of time. When I started off, I was not as rigorous with hiring. And of course, with a very early stage startup, you kind of so because we started from this kind of COVID thing, right? We were very lucky that people who are working with us were working for the right reasons.
Right,
They were excited to solve the problem. So in some sense, that solved my hiring for me. But then over a period of time, everybody makes mistakes in hiring, and so did I, and learned that some of these were avoidable mistakes. I could have had more rigor in my hiring, and I could have figured out how it is to be done. What you don't need to invent, you should copy.
absolutely.
I'm a big
Absolutely.
believer in that as
Right,
well. And so all these kind of things, instead of learning the ropes, it's much easier, much
right,
better to go and look at the best hiring practices and then bring them in.
right.
Yeah.
I mean, that when the moment you said best hiring practices and then you said uh, you should copy from the best references, the first thing I could think of was this Google's hiring policies as a whole. There's an interesting book. I'll probably send you a note on that. They basically talk about how they built the hiring engine from day one and how it evolved and how it
Very
is now
nice.
over
Very
time.
nice.
And there are some beautiful gems in that whole story, right? No one in Google actually, there are different parameters for things when you're hiring, but they often hire first for aptitude, or that weigh that a lot more than the skill, because their general belief is that if someone's really really smart, in terms of aptitude and picking up and learning things and have all the right directions in terms of how their value system is, learning a skill is not really a big challenge.
mm,
But if you over optimize for skill and not so much for aptitude, meaning, say, you just take someone in because there's a lot of experience in the field, but not really the other half, things can sort of become difficult. And
mm,
another important point that they mention is all of us, all startups, corporates, businesses, we spend a lot of resources in training people, any pretty much across the board, right? It's not just hiring and then running right on day one. There's a lot of training involved in the beginning, training at every time they have to get promoted, and this continuously you're sort of spending investment and resources in making them better. What Google believes is a little different. What they say is that if we spend at least half or one quarter of all the training budget in just making hiring more rigorous, like you said, they don't need the training after that. Like things sort of from day one, things go rocket speed. And I particularly loved that whole direction, and that changed methods within our system also.
mm,
Otherwise, the original team was a very organic team, like you said. The first few people in a startup, they don't really come together through hiring process. They sort of come together for good reasons because
mm,
they sort of believe in the common vision, and it's mostly vision match. And then someone comes in and they pick things that they're very good at and they end up doing. But as you grow, it's not the same.
mm,
You have to, like you said,
mm.
build process system, uh, add rigor, and sort of make it a repeatable, scalable model.
Yeah.
So another thing that I've sort of noted down as part of building the episode is this is interesting definition for startup, right? The definition of startup is is the act of finding a business model that is scalable, and can keep bringing more customers and keep delivering more revenues from existing customers.
Mm.
Until you find those matches, you're still in startup mode.
Yeah.
Like once you find that model and that becomes a thing, then you're off. Like that's a company, you can even spin it off, call it whatever,
Mm,
but you have a revenue model, you have a
mm,
business model, money is coming in, there is profit margin, then there's
mm,
net margins, things move forward.
mm.
In But for most of us, I think we are constantly seeking these new business models, right? So in your case for AlgoBio, are there a couple of ideas you're working on now in terms of business models, or you're exploring, you're still in that stage, or you think you found a match and you're going to go laser focus on it? So where are you in terms of that?
So there is a roadmap. Yeah. So right now there is Tapestry for NGS.
That's a product, it's
That's a
ready,
product.
right?
It's something we are super excited about, like with partners like Dr. Sudha and other NGS labs in India, internationally. We think that that is going to immediately bring down the cost of sequencing, and it is something we are super excited about. We are putting a lot of our sales and marketing effort on that.
So that's your
Yeah.
first
Yeah,
product
that is our
essentially.
first product, right? Behind that is this, Tapestry for a single sample, right? So the
Hmm.
assay that can test a large number of targets with a small number of reactions. So that is a partnership model. And here we have, some initial conversations happening with multiple partners, and that has started off,
Right,
and super excited about how that is going, right? And then there is the R&D, right? So we are building something we are super excited about, but too early to talk about.
right.
The vision that integrates all of these, right? So one concern that in, people can have when hearing this is, how is this like integrated, right? What is the thread that runs through all of
What's
these, or are
the
these just like, are you
red
running three
thread, right?
companies, right? Why are you running three
Right,
companies, right? So the vision that integrates this is that. Yeah, I mean, it takes a bit of time to say all of this out. There is like a medium term, there is a long term, and so on. So the medium term is that I have certain thesis about the market. Okay, so one of my thesis is that sequencing is a big deal, a much bigger deal than many of us realize right
understand,
now.
right.
Okay, it is, in fact, to put it somewhat more, in a big way, right? It is to the world of molecular information what the PC was to the world of information. Okay, sequencing is the personal computer of the molecular information world, and it is going to become ubiquitous in all kinds of situations there.
Right.
Okay, it will always be limited in some way, but right, it won't be a
At
point
least for
of
those spaces, it could be an everyday thing.
yeah. So it is going to be very important.
Right.
Okay, and sequencing is not about DNA. This is something very important. I think that it is playing out, and I think you and I possibly
Right.
know this, but
Right.
I think in the larger world, this has not yet come in. Just like the PC
Right.
was not about one use case,
Right.
right? Many different use cases mapped onto the PC, right? Whether
Right.
it is a spreadsheet, whether it is a document editing,
Right.
whether it is
PowerPoint and
paint and
whatnot.
so on,
Right.
right? Lots of things mapped onto the spreadsheet. In the same way, all kinds of molecular testing are going to map to sequencing. So DNA, RNA, proteins,
Protein.
lipids, metabolites, structural information, spatial. So of course, spatial, single cell, right? Temporal information, all kinds of things that you don't even think have anything to do with sequencing today are going to get mapped to sequencing, right? And each of these is going to be a different library prep and so on, right?
Right.
So that we are super excited about that sequencing thesis, and we think that making sequencing more efficient, whether in this kind of a tapestry for sequencing
Right.
manner or bringing the assay together, right? So the assay is now able to do some kind of a compression before sequencing,
Right.
then sequencing happens, and then we do a decompression
Decompression.
with software. So yeah, so again, right, so the vision here, right, so on the application side, this is kind of one direction where we are bringing these optimizations, right? Another thing, another metric that we are tracking for our R&D is sort of the uh, sophistication of the molecular computing that we are building, right? When we are building these assays, we have an internal measure which is some kind of a circuit complexity size,
Hmm.
right? And we are already in like few hundreds and so on, and we are
Okay.
seeing every year if we can move this up by a factor of 10, then
Oh,
in
wow.
4 or 5 years we would be at an order which is like millions or 10 millions or something
And
like
this
that.
is your
So
own internal
this
uh,
is an internal
North
thing
Star
that, and
metric.
as we are able to do, pack more and more compute
Hmm.
into into these assays, we believe that even more opportunities will open up, even more application areas will open up, which may not be directly related to reading information, but actually you will be able to do much more. It is not
Right.
yet a business story, this is more
The,
like a vision for
right.
the tech. Yeah.
Something that you would like to beat your own record year on year, sort of internal thing.
Yeah, so I mean, s- so the thing that I take away, right, is that the companies that do very well are companies that are in an area of science where there is a lot of room to go.
Right.
Okay, and so they can do this kind of 10x year after year after
Right.
year after year after year, so there is a lot of room to go and they keep going, right?
Right.
And so this is the axis on which I believe there is a lot of room to go, right?
Right.
We know what a cell can do, we know the circuit complexity of a cell in some sense, right? Huge number of reactions,
Right.
yeah. So uh, and it's all packed in a very small volume, it's all working, so we know that it's possible, right?
Right.
We don't know how to do it, right? And we may not figure out how to do it, but if we are building everything from the ground up, there is room to go, we know that, right? So it is not that 100 is the end of the road, it is not that
Correct.
1000 is the end of the road, it is not even that 100,000 is the end of the road, there is a long way to go. So we should be able to keep going,
Right.
and it is we may not anticipate immediately what we will do with that capacity, but then that has always been the case when
Yeah.
building any kind of
This
compute.
is
Yeah,
again reminds me directly of the Nvidia story, right? Like when they first built their graphic cards primarily for gameplay, rendering gameplays, and they kept getting better and better year after year, and then start building simulation software for across industries without even realizing which one of them will blow out into
yeah,
a mega
yeah,
app
yeah,
or a
nobody
killer
knows
app.
what to do with
Right.
compute until it is available,
Right.
and then somebody figures out something clever to do with
Right.
it and
And
becomes
finally,
something.
when
Yeah.
someone else figured out the GPT
Mm.
in the world of chatbots and
Yeah.
AI, now suddenly
And I
Nvidia
think, yeah, I
is
think
in the
partly
picture.
being deep tech means this to me,
Right.
that you find something where you can keep moving and
Right.
some arrow
Keep
of
improving.
progress. Yeah. And you know for sure that there is no hard bound there, right? You can
Right.
keep moving to an extent that seems ridiculous today. Yeah.
Geometrically speaking, a vector pointed in the right direction and keep going.
Keep going. Yeah.
That's
Yeah.
awesome. Now, coming when I said Nvidia, of course, everyone's probably already thinking AI. People who are watching this, right? Are you dealing with something in that space? Is AI impacting or directly coming into your work or products?
Yeah.
Where are we?
So to me it is obvious we are doing AI. Okay.
Right.
but it is not necessarily obvious to people who are not in
Right,
AI. Yeah,
right, right, right.
and because today AI has taken on some very specific meaning and so on, right? But for me, right, the kind of training I had, right, so AI and machine learning and then compression coding, these are
Fundamentally, all are
all essentially the
algorithms.
same.
Right,
Yeah, not
right,
just algorithms, right?
right,
There are algorithms which are not this,
right.
but these are essentially the same in a much deeper and like mathematically precise sense.
Correct.
Yeah. So yeah, I mean, there are books that explain precisely the connection between
Right.
machine learning, and so even diffusion models to a large extent map very well into some of the things that are going on.
And anything else for students? Now, that's another people who probably would be joining us eventually in a couple of years from now, or people who will turn into entrepreneurs themselves. You did speak to researchers who are in the tech side. We did cover some area there. What would you suggest people in this industry, the biotech engineers, biomedical engineers, who are looking to get into fields like these? So, what do you have to tell them?
So I mean, there are many paths. There is not one path. Yeah. Uh, but definitely one of the exciting things for me is this emergence of molecular engineering, bioengineering, whatever you want to call it. But yeah, I mean, really engaging with engineering with molecules and what you can build with molecules. So, in some sense, it's an old story, right? I mean, engineering with molecules is really just chemical engineering at some level, right? So, what chemical engineering was able to do was it was able to take the reactions that chemists were performing and perform it at the scale of a steam engine boiler,
Okay.
right? That
Right.
became the reaction vessel,
Right.
right? So scaling up the reaction, but it was still the same reaction happening in bulk. Okay. Then nanotechnology came along and we were able to work at a single molecule level, right? Single atom, single molecule level.
Right.
And biotechnology came, and keeping the reactor size, we were able to use enzymes to run much more complex, much more sophisticated
Right.
reactions,
Improve the
build products like
efficiency. Right.
insulin. Yeah, right. I think a new age is coming where we are. So think about what is possible inside a cell, right?
Right.
Inside a cell, it is not like inside a reactor. Okay, forget, of course, that's geometry is very complex and
Yeah.
so on, but even forgetting that, even forgetting that it is not well mixed. The point is that there are, like, it is really like the nanoscience case in the sense that there are individual, very exquisite reactions going on, but the scale at which it is going on, right, is also mind boggling, right? So there are like tens of millions or hundreds of millions of reactions going on, uh, and each of them is different, right, and all
Right.
happening simultaneously. So
Right.
that is something that we don't have a human analogy for, right, like human technology analogy for yet, right? But we are, in the process of sort of building up towards some of that, and learning how that is happening and building up some of that. So uh, people who are engaging with that kind of effort, I think there is a lot on the table there, right?
Right.
Yeah.
I've actually seen again slightly in the lines of bioengineering and then AI and then learning as a system. One of the researchers again in the US tried to map uh, EEG signals to thoughts, or at least if you imagine a image, or let's say you're imagining an apple or a banana, can you actually read your signal
Yeah.
live and recreate that
Yeah.
in real time?
Mm.
And did find success in
Yeah.
areas like this. I don't
Mm.
know if you've looked at this.
Yeah, fMRI based. Yeah,
fMRI
yeah, I've
and
seen some
fairly
of this. Yeah.
recent
Yeah.
Indian researcher, and stuff like that is boggling, right? You are actually thinking and someone can recreate your thought,
Mm.
and this is literally out of a sci-fi movie maybe just 10 years ago,
Mm.
when something seemed like it'll never happen
Yeah.
is already happening.
Yeah. Yeah, definitely. I think the brain is again like this fascinating unknown terrain where we've only scratched the surface
Right.
and a lot of way to go. Yeah.
When you really understand that therapies are guided by diagnostics, not the other way around, even though the amount of money you spend on diagnostics is much smaller compared to healthcare expense as a whole, it's a logical answer, right? Like something that costs you 10 or 20% of healthcare expense will actually really determine the success or failure of the rest 80% of expense.
I mean, I feel that if molecular diagnostics, right, or molecular testing can be improved 10x, it of course it's going to impact healthcare, but it's also going to impact, like you said, pharma and
Right.
uh,
Food
yeah,
and
drug
whatnot, quality and
discovery, like fundamental biology, right?
right,
Lots of different areas, and possibly biotechnology in unexpected ways. And so that, I think, wide spectrum understanding of the uh, in some sense of where the win is going to come from. So people who are very excited about biotechnology also will often not talk about molecular testing. But
right,
really, even if you're trying to build a biotechnology startup, where is the challenge, right? Often the challenge is that the startup has raised some money. Now they are in the lab, they are doing experiments, and those experiments are all you're pulling data out, right, from the molecular scale again and again and again, and you're exhausting your budget while trying to do
right,
that. And yeah, so I think building the infrastructure required is going to lift this entire kind of
right,
the ability of humans to work with biology in like very
right.
fundamental
And that
ways. Yeah.
infrastructure doesn't need to be $100,000 per instrument or doesn't need to be a couple of millions per establishment. It doesn't make sense.
Mm.
Like it's almost intuitive for any mechatronics like engineer when they see, if they really open it up and see some of these devices, most of them will realize that hey, it doesn't really need to be that expensive. Why is it still that expensive? And why is the access still so hard? As a company built out of India, building for India and for the world, that's also an interesting problem we tend to solve
So
very
do you
easily.
think regulatory capture is part of it?
No, it's just the way markets evolved in the West, right? Payers are involved, insurances are involved.
I see.
In most cases, healthcare is expensive. Today, healthcare is super expensive in the West. All of that sort of left room for these companies to stay at those pricing levels, stay within those very high margins for a very, very long time. But if you look at software today, in India at least, software or SaaS built out of India, they broke those shackles long back,
Mm.
right? The sales, your sales force doesn't need to be that expensive. It still is, but if you look at Zoho or any other India built software which are still operating really well in US and doing billions of dollars in revenues, they are not that expensive. They still deliver value, they still do what they're supposed to do, sometimes in even better in certain use cases. Now, the same logic can be applied to hardware, can be applied to biotech, can be applied to molecular computing, algorithms, and whatnot, right? We could build solutions that do better than what yesteryear's technology has done and can do it in a way that more people can access it very easily, very quickly. That's the good part of being in India at this
Mm.
time, where everything is sort of evolving, infrastructure is evolving, governments are getting slightly more smarter, and they're thinking about these things upfront. And I think we are at a good place. Like if we don't leverage it now, I don't know when else we can do a better job at it.
Yeah, exciting, exciting.
Awesome. So that with that, although the conversation is still unfinished, we will conclude this particular episode. And all the best for your venture going forward.
Thank you, Rohit.
And I'm waiting for some of
Wish
your
you the
public
same.
announcements to come through sooner.
Yeah.
We've been waiting for the good news for a while. Thank you.